Okay, guys, welcome to ZOO3649, Lecture 21. We're in the second half of the module now. And this is, as I keep saying to you, this is the business half of the module where the hard stuff, the stuff that makes the brain start really thinking. That's third year level Bachelor of Science stuff because, it's a Bachelor of Science. That means this one has to be capable of working. You cannot get a science degree if this one is not working. So do not be surprised that the module is becoming harder and harder and harder, which means that you will have to concentrate more and more and more. Okay, today we're going to continue with our Wright-Fisher population. And I just want to say, and I know I mentioned this in previous lectures, I just want to say to you, for your advice, this is going to be examined, okay? You will find questions about F-stats, you will find questions about Wright-Fisher population in this module, okay? So it's not something that you can ignore because it's too hard, okay? If you want to pass, there are going to be questions about this in the exam and in the test, so please make sure you know what you are talking about. Make sure you understand this, okay? So without any further hints, I will continue with the lecture. So we're continuing with the Wright-Fisher population, right? Remember, the Wright-Fisher population is idealized so that Sewell Wright could measure genetic drift, okay? He wanted to measure genetic drift, so it's idealized for that purpose. Okay, now Sewell Wright was a clever guy, and he figured out that if you have a Wright-Fisher population that is not one population, but maybe several different populations inside that Wright-Fisher population. Meanwhile, you think it's just one population, but the reality is there are several different populations inside that Wright-Fisher population. Then it will lead to very strange behavior of heterozygosity when you calculate heterozygosity in different parts of that Wright-Fisher population. And if you sound confused, if you're a bit confused, don't worry, we're going to unpack it in this lecture. Okay? And that's how Wright developed what we are calling F-statistics. And F-statistics, my dears, are very, very important to know. Okay? Because genetics, a lot of genetics and evolution is based around these ideas, these early ideas by guys like Wright and Fisher. So what do we do with this Wright-Fisher population? Say we want to, when we subdivide it into smaller populations, so if there's more than one population inside your Wright-Fisher population, strange things tend to happen, okay? So what we find is that if we look at different hierarchical levels, so there are different hierarchical levels of a population, remember? Again, there is a lot of, it comes back to the question, how do we measure genetic diversity? You know that question from 2544? Remember, we can measure it at the individual level, at the gene level, at the individual level, at the population level, at the species level. So there are different hierarchical levels that we can measure genetic variation. Okay? And this is basically what Wright is doing. He's actually measuring genetic variation or heterozygosity, which is one way to measure genetic variation. He's measuring that at different hierarchical levels, and he's taking averages at the different hierarchical levels, and they're not the same. Let us unpack some more, okay? And when they're not the same, it implies that there's some structure, okay? And that's how we know that this population is structured when hierarchical levels, the population at different hierarchical levels give you different values, okay? So the classic example, and it's still an example that we're using, is the Wright's blue and white flowers from the 1940s. So Wright, when he was a young guy, he went into the desert, and roughly in this area here that you see closed by the circle here. Okay, it's a small area on this map, but I can tell you it's a massive area. The area is around 30,000 square miles in the Mojave Desert in the United States. Because remember, Wright was an American man. And so he went into the desert and he found these flowers that are, the plants have, some plants have blue flowers, some plants have white flowers. And now you're thinking, this is a bit similar to Mendel, right? With his peas, some were blue and some were white. Well, it is similar in that. And what Wright, because he's very clever, what Wright figured out, and this is all he needed to figure out, was what was the frequency of the recessive allele in this landscape, okay, this Mojave Desert. So what he did was he went to, so wherever you see a number here, you see where my mouse is moving? Wherever you see a number here, like there, 0.573, or here, 0.339, or here, 0.106, or here, 0.004. Whenever you see a number on this map here, it's telling you at that place, right, what does he say here? That place covers, that one spot, say this spot here, where this number is, covers about 30 square miles. That's a big geographic area. And inside that 30 square miles, Wright went and collected data from 4,000 plants. Okay? And that's not 4,000 plants for the whole map there. No. It's 4,000 plants for each number you see on that map. So you can imagine how long it took Wright to do this, because that is a massive area to go and work out what is the frequency of the blue flowers, the blue allele. Okay, the recessive, and the blue is recessive. So because he's really clever, he only figured out the frequency of the recessive allele. In other words, which one did he figure out? He figured out Q, didn't he? So from Q, you can very easily do what? In a situation where there's only two alleles in the population. The white allele, which is dominant, and the blue allele, which is recessive. What was he able to figure out? If he knew Q, he can go 1 minus Q, and he can work out P, right? And then what can he do when he's got P and Q? Immediately, he can work out how many heterozygotes there are in that population. Expected heterozygotes. Okay, and how do you do that? You multiply P times Q times, you got it, 2. And that will tell you, so 2PQ, exactly. The Hardy-Weinberg expectation for the number of heterozygotes. So basically, what Wright was able to do, he worked out the allele frequency of the blue allele here, and then he basically, what he's got here is Q for all of these different populations. And what did he do? He measured, he minused it from 1, he got P, then he multiplied 2 times P times Q to get the heterozygosity of each of those populations where he took the allele frequency. And the other thing he did was he divided the population into 3 hierarchical levels. What do you think the 3 levels are? They are kind of marked out for you here. What's the very first level? The very first level is each population, right? This is a population of what? 4,000 plants. Each one is a population of 4,000 plants. So each one is a big population. So these allele frequencies are very accurate that he's taken, because he's taken so many samples. He's very accurate data, right? So each one of these is a population, right? So the first level is at the level of the individual, the heterozygosity within each population. Okay, the observed heterozygosity. Then, what is the next level up? The next level up is, you see, he's divided the entire region into 3, right? A western region, central region, and an eastern region. Okay, so then you can have heterozygosity and allele frequencies for just the western region. You can average here the west. You can average here the central, and you can average here the east. So that's the second hierarchical level, right? The west, center, and east. The first level, remember, is each population by itself. The second level is west, central, east. What do you think the third level is? You're going up now again, right? So what is the third level? Yes, you got it. The third level is the whole population, the total population. Okay? So that's the hierarchical. So you can take all these heterozygosities and take an average work out the average allele frequency for the whole population, and then work out P and Q and multiply by 2, and then you've got the heterozygosity for the whole population. So you actually have 3 different measurements of heterozygosity. Heterozygosity at the individual population, the region, west, central, and east, and then the total population, okay, for the whole Mojave Desert. Okay, so what did Wright figure out when he divided it into these 3 hierarchical levels? Look at this table here, guys. It's confusing a little, but not really. So don't look at allele frequency, because allele frequency is only for the recessive blue, okay? If you take that, you minus from 1, you will get the allele frequency for dominant white, okay, which is P. You times those 2 together with 2, and you get this number here, which is what? The expected heterozygosity. So for population, the one population, there's the allele frequency of Q, and here's the heterozygosity. So you know how you get from there to there. You take that one, minus from 1, multiply P and Q, multiply 2, and you get this one, this number here. Okay, I'm not going to explain that again. That's pretty obvious, and I've explained it now at least 5 times. Right, so what are you going to find? What did Wright find? So he took at the level of each population. So this is the heterozygosity for each population, basically for each of these different populations here, right? So this is the heterozygosity reading. He added them all up across every population in the landscape, whether it was from west, central, or east, it didn't matter. He added them all up, and he took a total and averaged it, and the total is 0.1424. You see that? That's if you take individual ones, add them all up, and divide it by the number of populations, and you get the average is 0.1424. Okay, great. So now what did Wright do? He went and said, okay, fine, I want to take the average just of the West. So he considered the West to not be 1, 2, 3, 4, 5, 6 populations. No, he considered the West to be simply one population. Okay, so he didn't care about the allele frequencies for each individual population. He wanted the total allele frequency of West, which is where? It's this here. Okay, so the total allele frequency, not for each population, but the all of West. The total allele frequency of blue is 0.5. He minuses this from 1 to get P, multiplied those 2 with 2, and you get the heterozygosity. So now he got a heterozygosity for all of Western. He got another heterozygosity for all of Central. And he got another heterozygosity for all of Eastern. And so what happens when you add these 3 up and you divide it by 3? Do you get the same as this number here? Where you took all of these and you divided by the number, you got this average. Are you going to get the same average? Well, Wright thought, yes, if my population has if my Wright Fisher population has no structure inside it, I should see exactly the same heterozygosity when I take the regions. But does he? He doesn't. Look, he actually finds a different heterozygosity, 0.1589, compared to 0.1424. So he was scratching his head thinking, Aish, I'm expecting to get exactly the same, but I'm not getting the same. Then he went to the next level, which is what? The total level here, which means that he took all the plants and he considered them to be coming from one population. Okay? And then he took the allele frequency for the one population and he worked out P and Q and he times by 2 and he got the heterozygosity for the entire population. And this heterozygosity was not the same as this heterozygosity for the regions. And it was not the same as the heterozygosity average over each population. So at the different levels, Wright was getting different values for heterozygosity. And he was scratching his head saying, Mar, how can I get different values? I should be getting the same values. Something is happening inside this Wright Fisher population. that is giving me different values for heterozygosity at different hierarchical stages. Guys, it's very important for you to understand this because it's going to be examined. Okay? It's going to be examined. And so basically, he figured out population differentiation in his Wright Fisher population that is happening thanks to genetic drift. Okay, so he worked this out from the hierarchical differences in heterozygosities that I've just explained to you. Okay, if you're not understanding, please go back and listen to it as many times as you need to get this, because it will be examined. Okay, and there will be a lot of marks involved. So don't miss out. Right, so Wright developed this idea into the idea of F statistics, which is a convenient way to show differentiation of populations at different hierarchical levels. And we are still using F statistics from 1940 something until the very present day. That is how ingenious the system of Wright was. Okay, so let's look at this box, and this is kind of another way to show you how a population has hierarchical stages. Like that population in the Mojave Desert, individual population, then regions, 3 regions, and then total. Well, here's another example. The box here, the square box, you see my mouse cursor? The square box is the total population. Okay? Each circle is a subpopulation. Okay? And each individual, you see each individual has 2 circles. Because why? You have 2 alleles. So this with 2 white circles is a homozygote. This with the black and the white circle is a heterozygote. Okay? So you can work out heterozygosity of the individual. So how many heterozygotes do you see? So how many do you observe, right? And the average number you observe. So that's the heterozygosity at this individual level, which is called HI. Okay? It's heterozygosity at the individual level within a subpopulation. So this one will have its own, this one will have its own, and this one will have its own heterozygosity individual. So what about HS? What is HS? HS is this here. The heterozygosity of the whole subpopulation, not of each individual here. Okay? No, not of each individual. This one is the heterozygosity of the whole subpopulation, this whole circle. So what is it? It's basically 2 times the frequency of P times the frequency of Q, but only inside this circle. So you can work out what is the frequency of Q, which is easy. It's the number of blacks divided by the total number of black plus white, the gene pool. Okay, that's Q. Then you minus from 1, you get P. Then you times 2, and you get the heterozygosity of this subpopulation. And then HT is the expected heterozygosity of the whole population. So basically you consider this whole square here. There's no divisions inside. Everything is one population. And then you work out exactly the same. You work out the how many, you count how many of the black alleles there are. Then you minus, you divide by the total gene pool. That will give you Q. You minus from 1, you get P. You multiply P times Q, and then you multiply by 2, and then you get the total heterozygosity for everything here, the total population. So now how are these 3 levels of heterozygosity, the individual, the subpopulation, and the total population? How did he turn these 3 into F stats? Let's have a look. So the first F statistic is a very important F statistic. It is the inbreeding coefficient. Okay, it's called FIS. Okay, why is it called FIS? FIS, because the F of I and S. What is I and what is S? You are talking. You're saying, Prof, what is I? What is S? Let's go back 1 slide. This is I. The HI, okay, I is the individual heterozygosity, and S is the subpopulation heterozygosity. So it's this one here. This is HS, and HI is the individual heterozygosity. So now let's go back to FIS. So it's the F that you get when you put the heterozygosity of the individual and the subpopulation together. Okay, so you get it by taking HS minus HI, so here it is here, HS is this one, minus HI, and you divide it by HS. And this gives you a statistic called FIS. It's a F statistic, and it's also the inbreeding coefficient. Okay? And this is the mean reduction in individual heterozygosity due to non-random mating. In other words, genetic drift inside a subpopulation. Okay? So basically, this is telling you how genetic drift increases homozygosity in the population. If FIS is a positive number, okay, it ranges between -1 and 1. So if it's from 0 to 1, if it's positive, that means you have a hell of a lot of inbreeding, a very high homozygosity in that population. That population is in big trouble because it's inbreeding, and inbreeding, as you know, is not good. Whereas if FIS is from 0 to -1, it's called negative FIS. That means it's negative inbreeding, and that's a good thing for the population. It means there's more mating with non-related individuals. So homozygosity is coming down in that population. Okay? And so that is a good thing for the population, and it's not inbreeding when FIS is a negative number. All right. So and remember from the last lecture, there's also going to be a very important question, something like, how do we model genetic drift in a Wright-Fisher population? Okay, and you will remember from last lecture that FIS, the inbreeding coefficient, is one way to model genetic drift in a Wright-Fisher population, because inbreeding coefficient increases in a Wright-Fisher population according to 1/2n. That is the holy grail number for genetic drift. Okay, 1/2n. So using the inbreeding coefficient is one way to model genetic drift in a Wright-Fisher population. Please do not let that out of your head. There are other ways which you will have to put to get full marks for a question like that. But FIS is one way. The other ways are what? The other ways are also 1/2n for the divergence of two Wright-Fisher populations. And the third way to model drift in a Wright-Fisher population is through the coalescent process, which we're going to get to in the next section. Okay, so that's question, how to model drift in a Wright-Fisher population, is a question that you can't memorize one slide for. Okay, you have to pull out of your knowledge of 3649, you're going to have to pull out, how are the ways that we model drift in a Wright-Fisher population? Okay, FIS is one of them. Okay, this is from the other lecture. Oh, that's the other. Oh, from the next lecture, another lecture down the line, that's the third way. You have to pull that all together and put it into one answer. Okay, no more memorizing. All right, so what's the second F statistic that Wright came up with? This is called the FST, and it's the fixation index. And FST, let me tell you, ladies and gentlemen, is even more famous than FIS. Because FST is the most published statistic in genetics. Okay? Since Wright came up with this in 1940 something, people have been publishing papers with FST, thousands, tens of thousands of papers. Okay? This is the most famous statistic in genetics, at least at the third year level, that you're concerned about. So how do we get FST? Yep, you figured it out, right? So we take the heterozygosity of the total minus from the heterozygosity of the subpopulation divided by the total. And let's refresh our mind again. HT is this one, the total for the whole square, minus from subpopulation, HS, divided by the total. Okay? So this is therefore the mean reduction in subpopulation heterozygosity due to genetic drift. Okay? FIS was the mean reduction in individual heterozygosity due to genetic drift. You must know the difference. You must know what it actually means in words. Okay? Remember, you want full marks for these questions. So it is actually also FST. That's why it is so published in almost every genetic study that's been known. It is a measurement of genetic genetic differentiation. How different can subpopulations become due to genetic drift? All right, and the FST, unlike FIS, it's not going from negative. It's going only from 0 to 1. Okay, so then that brings us to the last F statistic, the last fixation index, which is what? It is going to be FIT. And you've immediately seen how we get this, right? We take the heterozygosity of I, which is the individual heterozygosity, and the total heterozygosity, right? So we take the total minus from the individual, divide by the total, and this is the mean reduction in individual heterozygosity relative to the total population. Sorry about the spelling error. That should be a 2, right? Relative to the total population. Okay, and so it is also a measurement that combines the effect of drift within subpopulations and among populations. Okay, so how are the 3 FSTs related? There's a formula that relates all 3 of them, and that is this: 1 minus fit is equal to 1 minus FIS times 1 minus FST. Okay, that's a very, very easy formula. Okay, very easy formula to remember. Now, how much differentiation? Let's take Wright's F statistics, okay? And we want to know, what is the FST? This one here, this one. What is the FST? In other words, how much genetic differentiation is there among Wright's flowers, subpopulation of Wright's flowers? Okay, how much differentiation is there between Wright's flowers? So you know the formula for FST, okay? And I'm going to put in these values. So what are these values you're saying, Prof? What are these values? Let's go back to Wright's table. Remember, HT, FST is HT, the total heterozygosity, minus what? The subpopulation heterozygosity divided by the total. So let's go and figure out what those things were. Remember? Where is the subpopulation heterozygosity? Yeah, here's the table. Right? So here's subpopulations, okay? And the subpopulation average heterozygosity is this one here, HS, 0.1424, okay? And what is the total heterozygosity of the entire region? It is HT, this one, 0.2371. So now, 0.142 is HS, HT is 0.2371. And so now we go back to our formula. Here it is, 2371 HT, HS divided by HT, and we get 0.39 for the FST for that whole population of Wright's flowers. So how much differentiation is there in that whole population among subpopulations? Well, it's quite a lot. It's a huge amount of differentiation, actually, okay? Quite a lot of differentiation. 0.39 is a very high FST. Okay? Now, how do you know whether an FST is quite a lot of differentiation, very little differentiation, or moderate differentiation? Well, Wright helped us. Okay, Wright knew that we would be asking this question, so he helped us. He gave us guidelines for interpreting FST values. So if you get an FST that is very small, between 0 and 0.05. In fact, that's 0.05, excuse me, ladies and gentlemen, not 0.5, not 0.5, okay? It's the 0 should be here. Apologies for that. See where my mouse is? 0.05, not the way it's written here. So if you get FST that is low like this, then it implies little differentiation. If you get it a bit higher, between 0.05 and 0.15, Wright called this moderate differentiation. So the plants or whatever you're measuring are becoming quite different to each other genetically. Okay, and then above 0.15 to 0.25, so 0.25 is a quarter of the variation. Yeah, between the populations is in the data set is between the populations. That is implying quite a lot of great genetic differentiation. And if you get above 0.25, then it's a lot of differentiation. And what is our value here? It is definitely above 0.25, it's 0.39, and that's telling us that it is a lot of differentiation among Wright's flowers. Okay, and then let me very quickly, brush up on the island model, okay? And the island model of Wright tells us that here we have a Wright fisher population, okay, and it diverged into two different populations, okay, and in this situation where you have one Wright fisher population diverging into two, the rate of divergence due to genetic drift is, the clever ones have figured it out, 1 divided by 2n. That is the second way to model genetic drift using a Wright fisher population. Okay? So, what happens then? Once you diverge thanks to drift, and then you have migration, gene flow, between these two populations. Okay? They diverged, maybe they were isolated here. Okay? They diverged, and then suddenly they're not isolated here anymore, and they can do gene flow with each other. Right? Time has gone by. So now, what's going to happen in these two populations? You're going to have genetic drift, which is pulling the populations apart, like it is done here. You see? It has pulled these populations apart. And then you have migration, which is what? Bringing them closer together. The more genes they exchange, the more similar the two populations are becoming to each other. So genetic drift is doing what? Pushing them apart, the two populations, and what is gene flow doing? Gene flow is bringing them together. Okay. And so migration, gene flow is always interacting with genetic drift. In fact, you've seen this with your own eyes when you did the simulations during Practical 7. Remember, you did those simulations and you simulated genetic drift with an n dim size is equal to 12. Remember? And with no migration, migration 0, what was happening? All the alleles were being lost, right? And so the populations were becoming differentiated. So then what did you do? You increased migration to 0.001. Suddenly, one or two of those loci are not losing alleles anymore. It's keeping the gene flow, even though it was small, the migration was 0.001, but still it's bringing some alleles. It's not allowing drift to do its business and differentiate the two populations. Okay? Or differentiate or increase what? Homozygous genotypes within one population. Okay, that's what drift does. In one population, it makes things more homozygous, right? It gets rid of one allele. When there's two populations, it makes them more different. Okay, don't forget that. So, migration and drift are always they're like brother and sister, right? They're always in an equilibrium. Drift is pulling and migration is pulling in the opposite direction. And so they're always having a tug of war between them. And so, as you know from the prac, what did you do? You increase migration to 0.01. Immediately, what happens? You get less homozygosity. So you have fewer populations becoming extinct for one or the other allele, right? And then when you increase migration to 0.1, there's no drift cannot get rid of any allele from the population. Drift cannot make the two populations different. Drift cannot increase homozygosity within one population. Why? Because migration is there to bring the allele back after drift got rid of it. You see, that's why they're always in a balance and always having a tug of war. One is pulling one way to get rid of genetic diversity. The other is pulling the other way to increase genetic diversity. Okay, so that's basically what I'm explaining here. And so this equilibrium between drift and migration is actually described in what Wright called his island model. Okay, his island model where he's got F, okay, which is an F statistic equivalent to our FST, is equal to 1 divided by 4 times the effective population size multiplied by what is M here? The migration rate. Okay? Plus 1. So why 4? Because every parent has got 2 alleles, right? That means in mum there's 2 alleles and in dad there's 2 alleles. And so 4 alleles during the meiosis phase, 4 alleles are segregating. You as a child only get 1 of them from mum and 1 from dad. That's because they already segregated into different gametes, okay? But 4 alleles are segregating, okay? Which is why we've got 4 there. Okay, we multiply by 4 because 4 alleles are in a diploid population. Remember, a fisher population is diploid. That's why there's 4 alleles segregating at any meiotic phase. Okay, 4 alleles segregating, and then you've got the effective population size, which is the measurement of what? Force of evolution. No. If you're still silent and looking at me as though you have no idea what I'm saying, I suggest you go back to lecture 1 and watch every video again, and again until you come to a point where I ask you this question and you are like, obvious, prof. NE is a measurement of genetic drift. That's what I'm expecting you to say. If you're sitting there looking blank, you don't know what I'm talking about, go watch all the lectures again. Right, and then M. M, what is M? M is a measurement of what? The migration rate, okay? So you've got both forces of evolution, genetic drift, and migration inside NEM. That value NEM is taking drift with NE and M with migration, migration with M, and putting them together, okay? And that is the relationship between migration and genetic drift in a Wright fisher population, okay? And F is a probability of being identical by descent. Okay, it's an F statistic equivalent to FST. NE, the size of the breeding population in M is a probability of being a migrant. That means the migration rate. So then what happens if the population size is small here and the migration rate is also small? Okay, 1 divided by a small number is going to make F what? A big number. F is going to be nearly 1, right? 1 divided by a small number here, right, will actually give you almost 1 for F, okay? And what happens on the opposite when migration and drift, effective population size are large? Then what will happen is that the migration between populations will be so much that drift cannot make the populations different anymore. That the populations become identical to each other. In other words, the population size gets so big that genetic drift stops having an effect. Okay? And remember, when the population size is very big, drift stops having an effect. Okay? But to have genetic drift have no effect whatsoever, what size does the population need to be? It needs to be infinite. Exactly. Okay, so now you're getting it. All right, I will then see you for the next lecture, lecture 32, on the neutral theory. Another very, very exciting episode to look forward to in your journey through evolutionary genetics.